the recording, and if anyone has any problem with that, please let me know. And so the whole event will be recorded, obviously. And I would like, it is really my pleasure to introduce the speaker today. And his name is Daniele Vigolo, and he is a senior lecturer here at the University of Sydney. And he joined recently the School of Biomedical Engineering in actually almost 1 year ago, September 2020. And he is leading, co-leading with myself, the lab on a chip and organ on a chip cluster in the Nano Health Network. Just a little bit of history. He received a laurea in Italy in nuclear engineering at the Politecnico di Milano, and he received his PhD in radiation science and technology in 2010. He was also at Harvard University and Princeton University as a postdoctoral research associate. And in 2012, he joined also ATH in Zurich. So as you can see, the best universities in the world. And in 2015, he was also in UK. And his interest and main expertise is in the field of soft matter and complex fluids and biomicrofluidics. And today, I hope you will try to answer to the question, can we exploit microfluidics to engineer biology? So please, Daniele, the stage is all yours.
Thank you very much, Stefano. Let me share my presentation. Okay, so hopefully you can see my screen.
Yes.
Perfect. Okay, thank you very much. Hello, everyone. Thanks, Stefano, for the introduction. Yeah, so I'll try to answer the question today. So that's the idea of the talk. Before I go into the details of my talk, I actually wanted to just briefly introduce myself, basically extending a little bit the Stefano's introduction with a few of the topics I've touched in the past 5 years. So as Stefano mentioned before, I traveled here and there in the world. So I started in Italy, studying essentially complex fluids out of equilibrium, so in temperature gradients. But then I started to work with microfluidics right after with the Howard Stone, and that's where I really started to get involved with these small devices and to start to develop their potential. Once I moved back to Europe, back at ETH Zurich, that's where I start to apply microfluidics to biology. And finally, at the University of Birmingham, I really established my group, and I start to work in a number of different directions. And I think this is basically the key, the main advantage of microfluidics is really the fact that allow a multidisciplinary approach to a lot of different topics. And as a result, in terms of research, I've been also involved in a series of different problems throughout my career. I will show you in a minute. Finally, since last September, I am a senior lecturer at the University of Sydney, although technically I'm still not in Sydney, but hopefully soon. And I'm also co-chair together with Stefano, the lab and organ on a chip cluster of the Nano Health initiative, which I think it's a great place to exploit microfluidics for biological and health-related applications. So I've been working, considering my background in engineering, on a number of fundamental topics. So I start actually exploring microfluidics from a fundamental fluid dynamics and fluid mechanics point of view. So I've been looking at what happened essentially at the multi-phase flow, flowing in different flow regime through these small channels. And this is very fascinating because there are a number of phenomena that we discovered that were completely unexpected, such as this trapping of particles at the junction, for example, that happen at specific flow regime. But we also had a chance again, through microfluidics, to really look at what happened at interfaces. And this is another aspect of these small channels that give you a great advantage. In particular, we study the distribution of surfactant molecules during single droplet formation, which is, again, a key feature of microfluidics, for example. As I go along, you will see there's quite a lot of pictures, so I like to just give credit to all my students that actually did the work, so that's the reason of the picture there. I've been also studying foam. Foam is quite interesting in microfluidics because obviously they tend to have a completely different shape, and again, you can really apply, for example, in this case, we were interested in applying a rheological approach to the study of foams, and so we sort of create different ramps with different pressure, and we dynamically change essentially the behavior of the foam from different configuration to study rheological properties of the foam. But we also investigate these ultra-stable nanobubbles, which are fascinating topic on their own. Essentially, they are such a small bubbles that can be stable for weeks, potentially for months or longer, and they can be used to eventually stabilize or destabilize foams, but also other products. And this is really really an interesting phenomenon. As I was mentioning before, obviously, microfluidics give you the chance to really apply fluid dynamics principle essentially, but also material science principle to a much smaller scale. And this is a very versatile platform. And so I start to move my interest towards biology and biomedical application. And so we've been very interested in understanding, for example, the possibility to encapsulate bacteria for food application. And so I've been working on that for a few years as well. This is just a quick example where we can essentially incorporate in single and double emulsions different kind of bacteria that will actively help stabilizing the emulsion and at the same time represent a positive additive to specific food products. We also had a very, very interesting set of experimental results with proteins. So this, for example, is a very interesting collaboration with the University of Cambridge, where they are interested in studying protein phase transition and both liquid-liquid and liquid to solid phase transition. These proteins are typically pathological protein related to neurodegenerative diseases. And as you can see in the schematic here, you basically can induce a transition from just a pure protein solution through shear, for example, to form this beta sheet formation. This is really the main unit of amyloid fibrils that are responsible for Alzheimer's diseases, for example. So there is a clear link between a multi-phase fluids, if you want to look at this in terms of physical science, and the application of shear. So it's just, again, basic fluid dynamics, but there is a very strong health-related implication. So the biomedical application here is very obvious, and understanding this effect is really important. So as I mentioned, through microfluidics, we were able essentially to help them understand how the shear is applied and how to model this and to understand better this phase transition. Similarly, I was involved also in the study of a pneumatic isotropic phase transition of these amyloid fibrils, which are anisotropic particles. And again, you can use a concentration gradient, in this case, in a microfluidic channel, and really study the transition as it goes from a lower concentration to a higher concentration into a pneumatic phase. And again, this is another good example of multidisciplinary approach, in my opinion. Finally, and that's the main topic of today's talk, I was really fascinated by biomedical application of microfluidics. So I will be discussing in detail a couple of main results of my group in the next few slides, but I just want to show here some other things we've been doing in the very recent years. And we were really interested in incorporating biosensing into contact lenses, for example. And so we developed a series of different prototypes where we can measure a number of different parameters, again, tightly connected to specific disease or any way for health monitoring. Now, to move into the real topic of this talk, and to go back to the question in the title and to try to answer it. So the idea is really to try to apply an engineering approach to biology and try to see if we can somehow, by looking at biology and health from a very different perspective, again from an engineering perspective, if that can help. And so obviously my background is not technically in biology or medical science, so for me it's very ambitious to try to do something like that. But nowadays there are a number of different ways to try at least to tackle this topic and try to answer the question. So I want to focus on a couple of main approaches that are very popular these days, and I think these are the main direction where microfluidics can also represent a huge potential to help answer the question. So the first approach that I want to discuss is, of course, the possibility to reproduce essentially organ functionality on a chip. And so this is the principle of the organ on a chip device, but where we are going these days is really not only the single functionality of one organ, but a multi-organ functionality. And so we are really moving towards sort of a human or a body on a chip, but the real benefit, the real application, at least in my opinion, is really the possibility to have sort of a patient on a chip, and so really recreate specific conditions, specific situation where, for example, a drug can be tested and its effect can be studied on a multiple of organs, and so that will definitely help, for example, screening for side effects in terms of obviously different drug use that can be intakes in different organs, and that can be obviously beneficial, but can also be an issue to be investigated. I listed there just a few references, but as you can see, there are really a huge literature for the past 10 years. So this is a big topic in microfluidics, that's for sure, and there's a great potential to exploit there. The other aspect of microfluidics approach that can be used to really try to engineer biology is really in tissue engineering. That's obviously in the name itself, and also the principle where you can basically take some cells from a person, culture the cells, and then develop a new tissue to cure a disease or repair essentially the body is really fascinating and challenging at the same time. And the main challenge is really at this stage. So once you want really to create a proper tissue, a working tissue, you need to control essentially the cell behavior, and you want to control and program the cell behavior within the scaffold itself. So that's the very challenging part. And one of the reasons why this is very challenging is because obviously tissue in the human body is not uniform, and most of the time it's a very complex structure. So the scaffold needs to mimic this structure if it want to recreate the same tissue. I think a very peculiar example is really the Osteotendinous Insertion, essentially where is the connection between the tendon and the bones. And this is a very, as I mentioned before, a very complicated tissue. So if there is a damage to this tissue and you want to recreate this part of the body, obviously the complications are not only in the structure, but it's really the fact that there is a continuous change in composition, in structure, and in the properties of the substrate itself. And the reason for this is essentially because normally stem cells will take care of basically creating this, changing this specially graded material tissue in response of these properties that are changing locally in a matter of few hundred of microns. And if you want to recreate this, again, this is a good example of a very challenging problem to solve. So the control of the characteristic of the, in this case, the scaffold, the two-dimensional scaffold to reconstruct this part, for example, would be obviously very, very challenging. So I've shown you these two main approach, and I want to discuss two of our contribution in one in each of these two areas. So the first thing I want to discuss is the development of essentially a platform that gives us the possibility to study, in particular, deep vein thrombosis, but in general, it's a platform that represents a vein on a chip, and so it can be modeled to represent, again, microvasculature, and on that you can build up models for different disease if necessary. But I will focus on DVT. This is a topic that we've been working on for the past 4 or 5 years at least, and with at least 2 PhD students for the moment. The second part of my talk will be related to the tissue engineering part, if you want, and in particular, I will show you a new way that we devised to develop and control the characteristic of a biocompatible substrate. So the possibility to create these scaffolds where we can control locally the structure and the properties of the material to essentially pre-program the cells to develop on this substrate. So I will start first with the deep vein thrombosis part. So DVT is obviously a very serious disease, and nowadays it's even more relevant considering all the implication that, for example, the COVID vaccine has in the public opinion these days. I'm not discussing that today, obviously, but this is obviously a hot topic in these days. So, but besides the vaccine, this is something that happens quite often in the population, and we know that there are some risk factors connected to DVT, for example, prolonged immobility, and so when people is bedridden after surgery, or this is one of the main risk factors, but also pregnancy and old age are connected to DVT. I was fortunate enough at the University of Birmingham to have a set of collaborators really to tackle in a very multidisciplinary way this problem. So we work with cardiovascular experts, but also with computational experts to develop really an advanced model. And so Zoe first, that was my PhD student, she graduated recently, and now Hosam, they are both, they've been both working on developing this complex model, both experimentally and computationally. And what we really wanted to understand here is how we can connect essentially the properties of the vein itself, and in particular the flow pattern that develops in the vein with the formation of thrombus, and in general with DVT. So the reason why we're doing that essentially is that besides the fact that we know that there are risk factors for DVT, there are a number of questions that are that have no answer yet. For example, basically all the characteristics of the vein or the valve and the flow pattern, we know they are important, but it's not very clear how they actually affect the formation of thrombus. And I think this is crucial if we want to understand better the disease, but also if we want to try to prevent the disease and yeah, understand the mechanism behind this is crucial. So for this, we have to answer a few questions. So one is, can we manipulate the properties of the vein itself and the valve in particular, and can we monitor and discuss flow patterns? So for the vein part, well, the key part of this is not much the vein itself, but really the valve. So the typical position where a blood clot is formed is normally behind the valve, and so in the valve pocket behind the leaflet. And so it's crucial to be able to actually have passive valves that can move with the flow. And so we have control of the geometry, but also the elasticity of these structures. And to create this structure, we use in situ photopolymerization. We basically exploit a technique that is called stop flow lithography, where we can essentially fill the channel with photosensitive polymer, expose the polymer through directly the objective of the microscope and a photomask, and basically create a structure with the exact geometry that we want. But at the same time, because we can control the characteristic of the polymer we are using, we can also control the characteristic of each leaflet individually. And so we can create this small structure directly within the microfluidic channel. Now, the second challenge in this project is really evaluate the flow pattern. So we are working in microfluidics, and obviously the characteristic size are relatively small, which is an advantage, obviously, because we can control all the condition and we can have a very good optical access. But evaluating flow patterns at this scale, obviously, it's more complicated. So how do we do that? Well, obviously, we have requirements, and the requirement is that we need relatively high resolution, and these phenomena are pretty fast. And so we have to have also a very high temporal resolution. And the other characteristic is that typically we use tracers in the flow to evaluate the pattern, but these need to be really small to avoid to disturb and affect essentially the measurement that we are doing. So the technique that we developed in the past few years is ghost particle velocimetry, and the benefits of this technique are several. So one of these is that uses nanoparticles, and so they are very small tracers that don't disturb the flow. This must be below the diffraction limit, so they are essentially invisible. And we use just white light, so bright field microscopy to detect the flow pattern. The way we do that is by mapping the speckle pattern. As I mentioned, these are smaller than the diffraction limit, so you cannot see directly the particles themselves, but you see the optical effect of the light interfering with the nanoparticle themselves. By doing this, we have an easy way to basically extract quantitative information, and so flow patterns from our measurement, but we can also do three-dimensional velocimetry analysis, and we can essentially have information every about 20 microns within the channel, which is crucial for our investigation. Now, I think it's worth spending a couple of minutes on the GPV just to discuss how this technique works. So I mentioned that we are looking at speckled pattern, and a speckled pattern essentially is, you can think about as a signal that sort of follows the tracers. And what I mean with that sentence is just that the light is interfering with the particles themselves. So what you see and what you map, the speckled pattern, is not directly the tracers, but it's something that is directly connected with that. So if you're familiar with lasers, you will recognize this picture, and this is the standard speckled pattern that you will see from a laser. Obviously, the laser, being a coherent light, can originate speckles very nicely with a high contrast, and there are techniques that measure velocimetry through lasers, of course. But what we want here is really to have a three-dimensional scan within the sample, and because the laser is coherent, will give information throughout an average information essentially of the whole volume. And moreover, obviously the setup involved in lasers becomes a bit more complicated, and we want to be able to do as many experiments as possible. So we want to go with white light. Now, the problem with white light, of course, being at the other side of the spectrum, literally, is the fact that it's not coherent. And so you don't get speckles. So the interference of the light with the particle will just give up a random sum of contribution, and that will not form any speckle. And again, this cannot be used for velocimetry. So what we want, so the sweet spot will be something in between. So a semi-coherent white light will be the ideal goal. And so in that case, we will be able to have a velocimetry from a very small thin layer within the volume, which is very suitable for microfluidics, and it will simplify the setup. Now, in order to do this, we have to manipulate the numerical aperture of the condenser lens in a regular microscope. And by simply doing this, we can essentially reduce this this layer where the light is semi-coherent. But at the same time, we can also guarantee that we can generate speckle, and we can actually map the speckle. Now, this is the theory. How does it look in real life? So this is just a picture of a microfluidic channel with nanoparticles flowing in it. So as I mentioned, nanoparticles are below the diffraction limit, so you cannot see anything. The technique just involved the acquisition of several hundred frames, and once you take the median or the average of these frames, you get basically a very similar image to one of the frames. But once you subtract this and you do some image analysis, what you end up with, it's something that it's noisy, but you can clearly see that there is something going on. So you can clearly distinguish from a noisy background from a much more grainy contribution within the channel. And again, if your eyes can pick up the direction of the flow, so can obviously a software, and we can extract all the information in terms of direction and magnitude of the flow pattern. And that's exactly what we've done with our device. So what you're seeing here is the speckled pattern. Again, these are not the particles. The particles are smaller and much more diluted than what you see there. That's just an optical effect, but that's directly related to the flow field. And by mapping this, essentially, you can appreciate the presence, for example, of stagnant regions within these, but also the presence of vortices. And this is something that we investigated, because these vortices that develop essentially at the tip of the leaflet will grow, of course, with the Reynolds number, and so with the velocity magnitude of the flow, and they are somehow connected to the possibility for particle to accumulate, but also for platelets to be activated and eventually form a clot, which is what we want to understand in terms of DVT. But we were able to map different condition of different flow condition, and so map this characteristic of the flow in relation to DVT. Now, what is even more interesting is once we start to try to understand the formation of thrombus itself. So in that case, what we've done is try to mimic the formation of a thrombus by flowing larger particles. In this case, it's a 10 micron particle in our device. And what we noticed is a couple of things. So first of all, you can probably appreciate up here, you can see that there are the vortices I was discussing before, and they are clearly visible there. But what is even more interesting is that there is an accumulation, and quite strong accumulation, at the tips of the leaflet. Now, this is unusual because we know that most of the thrombus will be at least found behind the leaflet. So that's interesting that the accumulation points are the tips of the valve. More importantly, we've done that for different stiffness of the leaflet, and at the moment, what you're seeing here is an increase in stiffness. So basically, I have different concentration of float initiator, and the higher the concentration, the stiffer the valve. Both leaflets have the same stiffness in this case, But it's pretty obvious here how increasing the stiffness of the valve will lead to a much larger accumulation of the particles. And this is clearly seen here with both smaller particle and larger particles. Now, obviously, the complaint, if you want, here is the fact that this is just a purely aqueous suspension of polystyrene particles. This is not technically biologically relevant, but the idea behind this was really to point out the hydrodynamic component of this specific accumulation. So what we are looking here is purely what happened, driven by how the flow moves through these passive valves, and how the stiffness of the valves themselves affect the accumulation of particles. Now, to understand the biology, so we repeat the same experiment with whole blood, and in this case, we just fluorescently labeled platelets, but other than that, this is just regular blood from healthy individuals, and we saw essentially a very similar pattern. So again, the accumulation seems to be preferentially at the tips of the valves, and what is also interesting is that after the formation of thrombus, in this case, this is really an accumulation of platelet-based thrombus. This clot detaches, and so this is obviously one big issue with DVT. Obviously, the detachment of clot is the most problematic situation for DVT. Now, what is even more interesting is what happens if the two leaflets are actually not of the same stiffness. And again, this is probably this multidisciplinary approach that is playing around with this problem, because we ask ourselves, well, okay, the world is not always perfect. This leaflet cannot be always exactly the same stiffness. So what happens if one of the two is somehow more flexible than the other? And so that's what we've done here. And that's where things become even more interesting, at least in my opinion. So as you can see here, the leaflet on the left is much more flexible than the other. And because one is more flexible than the other, that will disrupt completely the symmetry of the flow. So you cannot see the vortices at the tip. And by doing this and having a pulsed flow, obviously, as it will happen in the vein, there is a strong accumulation behind the leaflet, which is where you normally expect to find a thrombus. And so probably stiffness, is clearly crucial in general in DVT. We know that, for example, older people have higher probability to get DVT, and that's also linked to the fact that the valves themselves become stiffer with age. But in this case, it's really the fact that they're not symmetric that really create this huge imbalance. And so, in a sense, the properties of the elasticity, essentially, of the valves and the flow pattern are affecting how the formation of the thrombus happens. Now, once again, we repeat the experiment with whole blood, and we saw a similar pattern. Obviously, the dynamics is slightly different, but again, once we have an asymmetry, we tend to see an accumulation no more at the tip of the valves, but behind one of the leaflet, which is somehow representative of an hydrodynamic effect. So from these, I think what is also very important to understand here is that because we are working with microfluidics, and typically microfluidics flow regime are obviously they are in it's a laminar flow regime, so it's easier in a sense to predict the behavior of the flow, but it helps also us to extract a number of information to build an in silico version of the in vitro model that we have seen here. And so we develop in parallel to our experiment also a computational method that can be representative of this. So I will not go into much details of the model itself, but this is a very interesting approach because we're not modeling the fluid as just a fluid, but each part of this model is composed by particles, essentially. So each small dot everywhere is essentially a particle, and we can control the characteristic of interaction of each particle with each other. And so we can define where the fluid is and where the solid walls, for example, so the properties of the leaflet are. And by doing this, we can allow essentially the valves to open and close and interact with each other. So contact is not a problem. And what we discover with this initial computational model is, for example, that there are preferential locations where a thrombus will grow. As you can see in this case, there are two sticky spots, so P1 and P2, and after some time, this is clearly the preferential location for the growth of the thrombus. But we also were able to monitor what happened to one of these thrombus after they detach, for example. And so the interaction with the subsequent valves, for example, depending on their size. There is clearly a lot of potential on this application. We are still working on developing more advanced model and approaches to even better understand this DVT in particular. So just to summarize this part before moving to the next one, I hope I show you DVT results, but more importantly, I think a platform where we can really develop a vein on a chip in this case, but if you want any blood vessel on a chip, where we can incorporate structure, we can control their geometry, their elasticity, and more importantly, we can really monitor how the flow affects this structure. In particular, because we discussed DVT, we can mimic thrombus formation, and I think the hydrodynamic aspect of the whole thrombus formation is really crucial, and it's important to understand even more in detail how we can understand and use this information to eventually make a better prediction in terms of risk factors. For example, taking into account the fact that valves can be asymmetric in elasticity, and that can be one of the risk factors directly connected to DVT. Now, the second part of my talk, as I mentioned before, is related to tissue engineering, and in particular, really on the manipulation of the properties of biocompatible materials that can be used as a scaffold, where we can grow cells and we can grow tissue, potentially following a predetermined behavior embedded in the material itself. So that's more or less the idea behind this project, keeping in mind that real tissue in our body are much more complicated and complex than standard uniform hydrogel, for example. So the reason why we want to manipulate the mechanical properties of these soft materials is because, beside biochemistry, mechanical stimuli are important factors that affect the development of a tissue and directly affect eventually how this tissue will behave. And if we are obviously considering to implant this tissue to repair the human body, the control of the structure of the tissue is crucial. And so we want to operate in terms of mechanical properties. We know from literature that a number of cellular behavior, motility, proliferation, adhesion, but also differentiation, if you're looking at stem cells, they directly depend on the stiffness of the substrate. This is a very famous paper where stem cells are cultivated on a uniform material of different stiffness. And as you can see here, there is soft, medium, and harder substrate, and there is a differentiation of stem cells in all this substrate induced by the stiffness itself. Now, I'd like to point out up here, just to give you an idea of the range of this stiffness, between 1 and about 100 kilopascal, we are moving essentially from a very soft brain-like tissue through muscle, through collagen bone, and so towards the formation of bones. This is the range of stiffness of interest for a large number of different cells. Now, what is interesting and what really struck my attention is that most of this research used different stiffness substrate, but uniform stiffness on the substrate itself. Whereas a gradient material is much more rare, especially a gradient that is actually sensible at a single cell scale. So that's what we were very much intrigued. So if you remember the example I mentioned at the very beginning of my talk in terms of the tissue connecting the tendon to the bones, this is a very small tissue, just hundreds of microns, where the structure is changing dramatically across that kind of size. So creating gradients is one of the ways that nature used to essentially guide the development of the tissue itself. So we were really fascinated by the possibility to create gradients and control gradients and use these gradients to guide cells. And so in order to do this, we wanted to develop a gradient material at a very small scale. So how to do this? Well, I got inspired essentially from what I've been doing during my master thesis. So back then, at that point was just the study of fundamental behavior of particles in a temperature gradient, so out of equilibrium. And so this effect, which is thermophoresis, applied to any suspension, gaseous and in liquid. So whenever you have a suspension, that can be particles, that can be ions in solution, that can be molecules in solution, as long as there is a uniform temperature, well, if these particles are colloidal particles, they will eventually diffuse by Brownian motion. But whenever you have a temperature difference across this solution, you will see a migration of the solute towards one of the sides. That can be the cold side or the hot side. That depends on the characteristic of the solute. But the reason for that is directly related to, as I mentioned, thermophoresis. And this is going to modify directly the mass of flux that develop. And so you obviously have a component, which is the standard Brownian diffusion component, as you can see there. That's just a fixed law. But there is another component that is directly connecting the flux of mass with the temperature gradient. Now, in this case, thermophoresis is obviously very similar. If you want to think to electrophoresis, we don't have an electric field. What we have is a gradient of temperature in that case. And so once the flux of mass is equal to 0, we reach a stationary state, and we have an equilibrium between concentrate. Oops, sorry. What's happening here? There we go. Sorry. So we have a equilibrium between the concentration gradient and a temperature gradient. If you see here, there is just the definition of thermophoresis and the dependence of this in terms of the Sorel coefficient. Sorry about that. There was an issue with my presentation. This is just how we define essentially the Sorel coefficient. Is normally positive when there is an accumulation to the cold side, because that's the most common accumulation direction. But this is not important at this point. So the most important part is really this balance between a gradient of concentration that is counterbalanced by a gradient of temperature. Now, this is true as long as you maintain a temperature gradient. But what if we can freeze this configuration and so keep this gradient there and translate directly a concentration gradient into a stiffness gradient, which is our final goal. So if we can do this, obviously we decouple the creation of a gradient and the choice of the material and the stiffness gradient that is connected to that. And so we can have essentially any material, which is very important when we are discussing tissue engineering, and so developing the right material for the right kind of tissue, and that's potentially very, very appealing. And so that's what I've been doing. So I developed that starting at ETH, and we look at the easiest possible sample to test. That was obviously biocompatible, but we also wanted something with a relatively low viscosity to develop this technique in a relatively quick fashion. So we started with sodium alginate, which again is a very well-known system. It can gelate in the presence of calcium ions, and we know for sure that the concentration gradient of sodium alginate will create a stiffness gradient. And so that was the ideal candidate for our initial test. So we've been doing this, and we developed a microfluidic device where we essentially can create our sodium alginate sample in the middle. We incorporate calcium carbonate nanoparticles. We'll see in a second why. But then we need to impose a temperature gradient to control the characteristic of the hydrogel. So to do this, we create a hot side by embedding a Joule heater by flowing essentially a low melting point alloy within the channel. And so that's, again, it's a great advantage of microfluidics because we can design any geometry for this heater, and we can just flow cold water on the other side. So this is how we impose a temperature gradient across our sample. Now, once we reach the desired gradient across the sample, bear in mind at this stage that the sample is still in liquid form. So we have all the time we want to create the gradient we require. We can then cross-link. And so the way we cross-link this is by reducing the pH in the sample, dissolving the calcium carbonate nanoparticle, and releasing calcium ions that will complete the cross-link process. And we do that through a membrane, a small membrane in the microfluidic chip. So this is the final result. Obviously, schematic looks much better than the reality, but the reality works. That's what matters. And again, we can control the Sorey coefficient depending on the sample we're using. In this specific case, sodium alginate at roughly slightly above room temperature, the Sorey coefficient is negative, so we have an accumulation. It's positive, so we have an accumulation to the cold side. But this is just a technicality. What is important is that we can extract this material and we can test it with AFM, and we can see the distribution of Young modulus across this material. So as you can see here, we normally work with about 500 microns, 600 microns fibers, and by probing along the across this tissue, we can see that if the material is not it's still produced within our microfluidic device, but without temperature gradient, we have a uniform stiffness across the material, and that's less than 10 kilopascal. But as soon as we impose a temperature gradient, we see the formation of a gradient of stiffness that is directly related to the concentration gradient we are creating. Now, it's worth noticing here a couple of things. So one is that we are changing also the average temperature when we are increasing the temperature gradient, but which is anyway not problematic. Of course, it's just a few degrees. What is more important is really to look at the gradient of temperature that we are imposing. So thermophoresis is proportional, the force, thermophoretic force is proportional to the gradient of temperature. And because we are working in microfluidics, we can impose very strong temperature gradients, although the absolute temperature difference is really small. So again, here we are talking about 0.5 millimeter material. And as you can see here, the highest temperature gradient is about less than 4 degrees per millimeter. So across this material, there was just 2 degrees difference. Nonetheless, we were able to induce a large difference in terms of stiffness across the materials. In this case, from about 30 to about 15 kilopascal. Now, again, if you remember the kind of range I was discussing in the beginning, so between 1 and 100 kilopascal means we are in the right range of stiffness, but obviously we are not, in this case, close to the bone side of the stiffness. We are probably more around the muscle stiffness in terms of a substrate. We wanted to explore a larger range, and we want to also demonstrate how this technique can be much more versatile. And so at the University of Birmingham, I've been working with my PhD student, Alex, and he developed this much more complex system. So in this case, we've been working with a different material. Again, with this technique, we are completely free to use any material essentially. And in this case, we choose to use gel and gum. This is a polysaccharide. It has a similar way to cross-link through the calcium ions, but the way we do this is by dispersing calcium ions directly in the solution and keeping the temperature above the cross-link temperature. And after we manipulate the structure and the stiffness of the material itself, we can drop the temperature quickly, and that will trigger the cross-linking, which is a very neat technique and very easy to implement. So the device, it's relatively similar to what we saw before. We have a channel in the middle with the sample. We have, again, a temperature gradient across with again, low melting point alloy and cold water. The device, of course, will require a Peltier module to control the temperature to induce cross-linking. Now, the benefit of this is that we can really explore a much large range of conditions. And as you can see here, this is just an example where we can impose a larger temperature gradient. And by using, again, I'm saying a larger temperature gradient, we are still discussing 3-4 degrees across the material. We're not discussing much more difference in temperature, but again, the gradient being the material so small is quite strong. And by doing so, we see that the profile follows exactly what is expected from the theory of thermophoresis, and it follows exactly an exponential decay. So this is the distribution that the concentration will have, and we know that the concentration is directly linked to the stiffness, and so we can monitor that what we are actually looking is the same thing. So the way we perform AFM measurement is essentially to take at each location a map of several position and then averaging the value. So you can see here the distribution on 4 different locations, for example, of the stiffness. Now, as I mentioned, we know that there is a linear behavior between concentration of the material and stiffness, because we prove that by essentially testing different materials with different concentration, uniform material, and linking directly stiffness to concentration. And that gives us the possibility to basically calibrate this curve, not only with the Young modulus extracted from the AFM, but directly with the local concentration. So we know essentially at each point what's the local concentration of the gel and gum in this case. So in this specific experiment, we started with a 1% concentration of gel and gum. We end up with a material that reaches 1.25% on the stiffer side and just above 0.5% on the softer side, so that we can both connect the concentration and the stiffness. Again, the overall idea is really to understand how and characterize the system so that we can manipulate this. Moreover, we had a collaboration with the University of Cambridge, particularly with the group of Professor Knowles and with Dr. Shen, who just moved as well at Sydney. So she's in the School of Chemical and Biomolecular Engineering right now. And we developed this technique where we can study not only the stiffness, but we can link directly stiffness and porosity of the material. Now, the final goal, of course, is to develop a scaffold for cells, and cells are very sensitive not only to the stiffness, but also really to the structure of the material. So porosity is definitely a very important characteristic that links directly focal adhesion points of cells and their development and their behavior. And so mapping this and being aware that we can control not only the stiffness, but also the porosity, this was very important to us. So we were able to characterize both things. At the same time, this gave us also visually a confirmation that we really have a different concentration at different location. Not that we needed an additional proof, but this is always nice to see a real picture of a tightly packed location at the stiffer side and a much more larger porous size and so on, a lower concentration overall at the softer side. Now, this is the way we develop the material. But again, the goal is to develop a biocompatible material to be used as a scaffold. We have to prove that this is actually possible. And so we do some cell study. In particular, we were interested in looking at osteoblast growth on this material. Because of the stiffness range, you can see here, with this material, we can reach about 70, 80, but depending on the material, we can reach basically between about 10, 20 kilopascal up to 100 kilopascal, which is a stiffer enough range to really affect the behavior of osteoblasts. Again, towards the 100 kilopascal, we are moving towards the collagenous bone range. So that's where the choice of osteoblast came into place. And so we culture osteoblasts, and the schematic of our approach was to seed a very low concentration, very low number of cells in the beginning on our material presenting a gradient of stiffness in this case, let them grow for about 7 days. And that's where we start to appreciate how proliferation and migration are affected by the gradient. But then waiting more than 15 days, we start to see how the maturation of the cells and the fact that obviously osteoblasts want essentially to form bones, and so they will start to mineralize the substrate, and we can appreciate that as well. So this is the picture of the live and death assay. So in day 1, as I mentioned, very low concentration of cells, but after 7 days, you can clearly see that in a uniform substrate, the cells are spreading everywhere in a more or less uniform fashion, whereas in the stiffness gradient material, we have clearly a preferential direction where cells move towards the stiffer area. Now, we can quantify that, and we know that there is essentially no difference in terms of cell viability, but the large difference is, of course, in the preferential direction where cells tend to accumulate, and so towards the stiffer area, which is, again, expected considering that these are osteoblasts. Now, the last and interesting part of this experiment was the fact that osteoblasts can mineralize the substrate. So if you leave enough time, they will start to release mineral directly on the substrate, building blocks to eventually create bones. So if you see the picture here on the right, this is a sample after 30 days. And you can probably barely see that there is our strip of material in the middle, but there is definitely a large amount of mineral deposits. So this is the sign that the osteoblasts are healthy, and they are actually behaving as expected. So we did 2 sets of experiments. In this case, we use normal medium and osteogenic medium. The osteogenic medium is just a medium that is not only a nutrient for the cells, but it's also promoting essentially this mineralization. And we noticed that in both cases, our gradient material is actually promoting mineralization towards the stiffer side as expected. And we quantify that by XRF. So this is the results we obtained. So if you see here on the left-hand side, this is just a picture of the material under the XRF, so in a vacuum. And we can map essentially the whole material for different minerals, in this case, calcium and phosphorus. And along the gradient direction, you can probably appreciate from this picture that there is actually a gradient in the mineral concentration, but I think it's easier if we look at the plot on the right. So now the first 2 plots here, these are using normal medium. So there is no, the medium is not increasing essentially the chance to form minerals. And after 30 days of culture, as you can see in yellow, we have basically the control experiment. These are materials that have a uniform stiffness, and they have also a uniform distribution of both calcium and phosphorus. But as soon as we are in the presence of a stiffness gradient, these are the light blue and the purple line here for calcium and phosphorus, we have a gradient in the concentration of these minerals that is following exactly the stiffness gradient that we impose. So, and this is clear with normal medium. It's even more obvious with osteogenic medium. As I said, osteogenic medium is sort of increasing the chance to form minerals, and as you can see, in terms of weight percentage, the absolute number is higher for osteogenic medium, but the behavior is substantially the same in both cases. So that tells us that we can embed cues, mechanical cues or structural cues within the material, and we are expecting, and as expected, the cells respond to these cues and they behave accordingly. So if the idea is really to promote the formation or the migration of these cells towards one direction, this is one way to go. So to summarize this second part, I think hopefully I show you again, hopefully a new technique where we can really control the characteristics of a biocompatible material. And what is really important is that we can do that independently from the choice of the material itself. In particular, we see that we can create a gradient of both mechanical properties and porosity controlled by thermophoresis. And this permits us what I think is the main message here is really to pre-program the material itself to pre-program how the cells will behave on that material and how they will eventually develop and form tissue by simply controlling the characteristic of the material itself. In the particular case, we've noticed that osteoblasts, as expected, migrate towards the stiffer side, and they also mineralize in that direction as well. Now, before I conclude, I want to obviously acknowledge who actually did the work itself. So my group, my funding in the UK, where I did most of the research I've shown you today, and really to finish in the last slide with really the initial question and hopefully to try to answer finally the question. So can we engineer biology? And this is my opinion, but I think, yes, I think we can do it, but clearly we need a multidisciplinary approach. And so I think this is the right situation to mention that collaboration are really crucial. And so please, if you're interested in any of this, and if you want to join forces, please let us know. And as mentioned before, Stefan and I are co-chairing this Lab and Organ on a Chip cluster of the Nano Health initiative of the Nano Institute, which I think is a brilliant initiative where really all this interdisciplinary approach can work together around microfluidics and lab and organ on a chip to really hopefully move these kind of things forward. And with this, I thank you for your attention, and I'm more than happy to answer any questions.
Thank you, Daniele, for this fascinating talk. So I'm opening for questions. If anyone has any question? You can raise your hand if you want, or just ask this question directly. Okay, maybe I'll break the ice. What do you think in terms of the first part of the DVT? Can you use your methodology to diagnose the risk factor, or more actually to engineer a new tissue that can be implanted later on? What do you think is the vision for microfluidics in that context?
Yeah, thanks. I think that's one of the really the most important thing, especially for DVT. This is one of the goals that we had since the beginning. So yeah, okay. So I think again, being an engineer, not having direct knowledge of how a medical practice works, in the beginning we faced this kind of problem in a very naive way. But we had discussion with medical doctors, and we understand better how the whole process of DVT, of detection and sort of prevention and the whole process work. And what is really important is really to create a sort of a table of risk factors, and this is where really we wanted to contribute at some point. So trying to understand how the different aspects, in our case, the flow pattern, but also the characteristic of the valves, for example. So if you can have pre-detection and you know that you have a condition where the valves, for example, are are more or less stiff and they're not symmetric, that can be a factor that can be quantified. And so really the aim for this standardized approach, because at the end of the day, microfluidics give you really the chance to have an environment which is fully controlled, where you can really change each parameter one by one in theory, and then create a table essentially of risk factors. And that will be very powerful, in my opinion, also to transfer into healthcare practice, for example, because we'll give really a practical way to quantify things and see how this will affect the final output, for example, of DVT. Now, similarly, you can do similar things in other fields. So in tissue engineering as well, you can quantify. I think the quantification that is possible through microfluidics is really a key. And again, it will enable essentially the well, you can create a series of experiments that are fully controlled, and that will give you the possibility to really test different conditions and extract possibly detailed information on how to create a new tissue or how to quantify the risk of a disease. These are all possibilities.
Yeah, thank you. Do we have somebody else? Any other question? So if nobody has a question, I actually would like to ask you something else. You talked at the very beginning, actually, you mentioned at the very beginning that microfluidics can be used to engineer organs. And that's kind of I understood your vision to have the entire patient on a microfluidic chip, basically. And obviously we know that our organs are three-dimensional, and the functionality of the organ seems to come more from the whole full three-dimensional structure rather than a two-dimensional part of it. And can the microfluidics platform adapt to the three-dimensional structure and functionality, or is limited to the two-dimensional part of it? And how can you link the two? Because the functionality that you can probably engineer on a two-dimensional tissue or structure probably are different from the actual organ. Is that true?
Yeah, thanks. This is a very, very important question, and it will take probably another hour of discussion to answer, and probably we will not reach a final answer. I'll try to give a 2-minute version of the answer. Well, I think you're absolutely right. So real organs, exactly as I mentioned, real tissue are obviously complex. There is a three-dimensional nature that it's unavoidable. But there are some specific aspects that you can reproduce. So I think you need to sort of decouple the advertising way to discuss microfluidics from the practical application. So when you say organ on a chip, you might advertise as a complete organ functionality, where the reality is that we can break down many different parts of the function of one organ and reproduce those. And those are definitely important. They are definitely linked and directly, you can directly use the results from those experiments to tailor, for example, drug screening. That's definitely something that has been done and it's ongoing. But again, you cannot forget that the full organ is a much more complex system. So we need definitely to link the two words. So there is a macroscopic three-dimensional biological approach, and microfluidics. This kind of microfluidics is basically the sort of a 2D approach, but there is a lot in between. So there's a lot of work in terms of organoids and anything in between, where you can start to implement three-dimensional structure and let essentially the cells construct this three-dimensional structure, or you can guide the cells to create the kind of structure you want to study. The control is much more complicated, obviously, in this three-dimensional structure, but I think there is a clear link. So once we understand fully the two-dimensional, we are not, moving from that to the three-dimensional will be more complex, but it's not you know, it's directly linked. So the information we gain from the 2D are still very, very important.
All right, thanks.
I think there was a question in the chat. I was looking.
I think it was Greg saying that he had to leave and he was really happy to. No, I cannot see any.
Michael was asking about sickle cells. Sorry, this is a direct message to me. That's a good question. That's why you cannot see it. Okay.
Oh,
yeah. So yeah, Michael Morris was asking about sickle cells and the connection essentially with venous thrombosis, and if I have tested them in this. I think this is a great question, and we actually didn't do that. But what we discover as we are working with this platform is that we have way more questions than in the beginning. So there's so many things we can actually do with this platform. So which is positive in one side, in one sense, but obviously it's kind of moving us a little bit all over the place in terms of different things that we can do. And this is definitely one of the things we are interested in. So we are slowly moving into adding more and more biology into our in vitro system. And I think that's, at least for us, was a good way to approach this, because we know better the physical science of this sort of problem rather than the biology, and then adding biology adds a lot of complexity. So that need to go a bit more slowly when we are confident that the whole system is working, and that's exactly what we are doing these days. So we are developing a much more advanced model where obviously all these components, again, biochemistry and not only fluid dynamics and mechanical properties obviously play a huge role. So the effect of, you know, cells lining within the vein, but also different stress in terms of vein behavior and their effect on the thrombus formation. There's a number of different things that we are looking at, and sickle cells definitely is one of the things in the list. So that was a great question. So I think that's yeah.
Okay. Any last question? Okay, if not, I would like to thank Daniele for this journey. It's really fascinating and interesting. And I would like to give everyone have a good night. And I believe the same to you, Daniele. Thanks a lot.
Thank you very much.
Bye.
Bye-bye.